Now, to get more clear concepts concerning the level of saturation and how it affects the fluidity of the cell membrane, let's go back to our two cell types, okay? So, permit me to clean this part of okay, fine. So, in these two types, different types of cells, we're going to consider cell type A to have 100 different, there are millions of phospholipid structures in a particular cell. Okay, taking one cell, there are millions of phospholipids. But in our illustration, we're going to consider cell type A to have phospholipids of 100, okay, 100 phospholipids. And the same to apply to our cell type B. In cell type B too, there are 100 phospholipid structures. Fine. Now, what is different is that in cell type A, out of the 100 phospholipid structures we have in here, 40 of them have saturated fatty acids. Okay, so in this structure that we have on the bottom right now, you know that a phospholipid has a glycerol backbone and then 2 fatty acid chains. So out of the 100 phospholipids in cell type A, there are 40 with saturated fatty acids. What are you trying to say right now? So out of the 100 phospholipids with saturated fatty acids, let's try to put it this way. Phospholipids with saturated fatty acids in here are 40, whereby phospholipids with unsaturated fatty acids, okay, take up the remaining 60. That is what is peculiar to our cell type A in our illustration. Let's go to cell type B. In cell type B, out of the 100 phospholipid types that we have in here, okay, out of the 100, we have 60, okay, having saturated fatty acid types. So what are you saying in cell type B? Out of the 100, phospholipid types with saturated fatty acids are how many? They are 60. But the phospholipid types with unsaturated types of fatty acids cover up the remaining 40. Now, the question we ask ourselves right now is that how will the differences in the levels of saturation in our cell type A and cell type B affect the fluidity of the cell membrane? Now, this is where we come back to a rule that you have to bear in mind, okay? The rule says that increasing the number of saturated fatty acids over the unsaturated fatty acids in the phospholipids in the cell membrane decreases the fluidity. Let me try to say it again. What we are trying to establish right now is that increasing the number of phospholipids with saturated fatty acids over the phospholipids with unsaturated fatty acids decreases the cell membrane's fluidity. So looking at our cell type A and B, realize that in cell type A, the phospholipids with saturated fatty acids are 40, but the ones with unsaturated fatty acids are 60. But in cell type B, the phospholipids with saturated fatty acids are 60 over unsaturated fatty acids being 40. And going back to our rule saying that the more you increase phospholipids with saturated fatty acids over the ones with unsaturated fatty acids, the less the fluidity. So what do we say now? Which one will have a cell membrane with a higher fluidity? Now you're considering the cell membrane with a higher fluidity. So a cell membrane with a higher fluidity in terms of levels of saturation will be based on the cell membrane that has phospholipids with a higher unsaturated fatty acids. Is that okay? So which of them? Obviously it's going to be our cell type A. Now cell type A, the unsaturated fatty acid phospholipid types are more than that of the saturated types of phospholipids, making it have a cell membrane with a higher fluidity as compared to cell type A. That's what it means to say that the level of saturation of the phospholipids the level of saturation actually of the fatty acid in the phospholipid in the cell membrane affects its fluidity also. So that is that. Okay, now, so guys, we know that the fluidity of the cell membrane is affected by a lot of factors, okay? And now we've come to discover 2 of them. Get to know that these are not the only factors affecting this special fluidity property of the cell membrane. But now coming down to this far, okay, concerning our phospholipid structure, we now know that the length of the fatty acid binding to our glycerol backbone, together with the level of saturation of these fatty acids, influence the fluidity of the cell membrane. Fine. So take note of these concepts. Now, let's come back to what is actually binding to our carbon 3, okay, the hydroxyl ion of our carbon 3. That is the next thing that you're going to do. There are a few concepts to cover concerning our phospholipid structure, so we can leave it alone, right? Now, so what is actually binding to carbon 3? The molecule binding to carbon 3 is the reason why we have phospho, the prefix phospho in the phospholipid name, okay? So what is this molecule at all? We call this molecule a modified phosphate ion. What is the name of this molecule? You see, the molecule is a modified phosphate ion. So now we ask ourselves, what is the phosphate ion and what does it mean to say there is a modified phosphate ion? A phosphate ion, for which we normally call in biochemistry phosphate group, is just a molecule that has a central phosphorus ion surrounded by oxygen ions and an atom. Okay, fine. So this is simply our phosphate ion. So what does it then mean to be modified, and why do we have to respect a modified attaching to our phosphate ion? Because just adding a phosphate ion to this molecule that we have on the board. Now, there is one thing to tell you. The glycerol backbone together with the fatty acid chains even form a molecule on its own before attaching the phosphate ion, okay? It's even a molecule on its own. We call this molecule, we find it a lot when you are dealing with lipid digestion and absorption, okay? We call this molecule diacyl glycerol. Diacyl glycerol. So what is diacyl glycerol? Diacyl glycerol is simply the glycerol backbone together with the two fatty acids. Fine. So now, how are we transforming our diacyl glycerol into our phospholipid structure? This is where we said that we just need to attach a modified phosphate ion to what? The carbon 3 of our glycerol backbone. And we are saying that, what does it mean to call it a modified phosphate ion? Why do we have to respect the modified term? Okay. Now, we are saying that this is our phosphate ion and it alone binding to our diacyl glycerol compound doesn't make this molecule our phospholipid structure. Okay. This binding to the diacyl glycerol transforms this molecule rather to what we call phosphatidic acid, but not our phospholipid, okay, or we call it phosphatidic acid or phosphatidate, okay, fine. So what is phosphatidate? Phosphatidate is our diacyl glycerol, which means a glycerol backbone with two fatty acids, okay, attaching to a phosphate ion. That is the meaning of phosphatidic acid. So then, what is therefore a phospholipid? A phospholipid is a diacyl glycerol having a modified phosphate ion. So then we ask ourselves, how do we then modify our phosphate ion? So to modify phosphate ion is just simply adding up the molecule. So a modified phosphate ion is a phosphate ion that has an extra molecule attaching to it, okay? An extra molecule attaching to it. We're going to use this star to represent those extra molecules that can bind to phosphate ion to render it modified. And remember that concludes the phospholipid structure, because you having this alone together with the diacyl glycerol is phosphatidic acid, as we are saying. But when this extra molecule modifies our phosphate ion, that is where we have the different types of phospholipids that we have. So the molecule that will bind to our phosphate ion to get it modified will determine the type of phosphate ion, phospholipid that we are talking about. Okay, we will talk about the types of phospholipids later in our lecture. And get to know that we are trying to have it in our mind that only phosphate ion binding to our diacyl glycerol doesn't complete the phospholipid structure unless we modify our phosphate ion with different structure altogether. So the structure can be choline, can be ethanol, amine, can be a structure called serine, sometimes a structure called inositol. These structures add up to modify our phosphate ion to differentiate between the different types of phospholipids that we have. Is that okay? So that is fine. So what we are trying to say right now is that the phospholipid structure is made up of a glycerol backbone binding to two fatty acid chains and a modified phosphate ion. Fine. So this is our complete structure of our phospholipid. Okay, now guys, then it is time to make our phospholipid structure small, okay? Remember before we came down here to this little concept, we said that the phospholipid structure is a lot in the cell membrane. For that matter, assume you want to illustrate 10 out of this cumbersome structure we have in the body representing phospholipid. You can imagine how much time that is going to cost us, right? So because of that, you want to deduce a much more smaller structure out of this huge molecule to represent it wherever that you draw it. Is that okay? So let's try to do it right now. So phospholipid can be made small in this manner. We're going to consider the phosphate ion, the modified phosphate ion, to be our head group. Okay, so this that we have in the body right now is going to be our head group. So let's try to bring it here. So we're going to consider that to be our head group, whereby the other sections, okay, this diacyl glycerol putting together the glycerol backbone and two fatty acid tails, okay, the fatty acid chains that we have over here to represent our tails of the phosphate. So let's try to put it over here. So this is our head group represented by our phosphate ion, and then one tail, another tail. Fine. So this is what we are deriving out of our huge phospholipid molecule, but you can even make it much more smaller. Let's try to stand it upright, okay? So our head is going to be pointing up, then we having one tail, another tail. So guys, this is what we're going to use to represent phospholipid wherever that you draw it. So in drawing about 10 of phospholipid, this will be a little bit quicker, right? Instead of using this cumbersome structure. Is that okay? So that is that. But before we leave the phospholipid structure alone, this outlook that we have representing phospholipid has two distinct sections, two distinct important sections, okay? Because the head group, which is represented by our phosphate ion, has some charges on top of the oxygens that it bears. It is making the phosphate group or the phosphate ion the hydrophilic domain of our phospholipid. What's the meaning of hydrophilic? Hydrophilic means that it loves water. You know, because it is having some charges on top of it, it is a charged molecule. And charged molecules are polar. And polar molecules dissolve in water. And then in chemistry, molecules that dissolve in water are tend to be water loving. And in more scientific terms, you see that they are hydrophilic. So what are you trying to say right now? We're saying that this head group, okay, this polar head group, or this phosphate ion represented by our head over here, is going to be the hydrophilic domain of our phospholipid structure. Okay, the hydrophilic domain. Whereby, because these tails that we have represented by the diacyl glycerol, there are no charges on them. Because of that, they are uncharged and therefore non-polar. And being non-polar, they don't dissolve in water. So because they don't dissolve in water, they tend to hit water. So the tails that we have over here, they are the water-hating, okay? They are what? The water-hating section or domain, okay, of our phospholipid structure. And in more scientific terms, we say that they are the hydrophobic. Hydro means water and phobia means to hit. So hydrophobic domain, okay, of our phospholipid structures. Is that okay? So guys, this is what we're going to use to represent our phospholipid structure. And now we've come to understand that because this head group or the phosphate ion is charged and therefore loves water, it's going to represent the hydrophilic domain of our phospholipid structure. And then because the tails are uncharged, therefore non-polar, and therefore water-hating, they are representing the hydrophobic domain of our phospholipid structure. So bringing it here, our phospholipid structure has how many domains? It has two domains, right? The head group, represented by the phosphate ion, the modified phosphate ion, is our hydrophilic domain. Whilst the what? The two tails here represent the what? Hydrophobic domain. Please take note of these two distinct sections of the phospholipid structure because they determine how the phospholipid structures are arranged in the cell membrane. So I want you to take a close look at it. So now, what you're going to do next is you're going to take our cholesterol molecule, know the typical orientation of the cholesterol molecule, and that simple structure you can deduce out of it. Okay. Awesome. Okay guys, so now let's consider the typical structure of cholesterol. Right? As we discussed, we want to know the typical look of cholesterol, that simpler, much more smaller structure we can deduce out of it to represent it in drawing the foundation of our cell membrane. Is that okay? So how does cholesterol look like? How does our cholesterol molecule look like? In the structure of cholesterol, there is this nucleus called steroid nucleus. Remember when we were talking about the structure of phospholipid, there was this backbone called glycerol. But in cholesterol, it's not a backbone but a nucleus, and we term it as what? A steroid nucleus. So how does this steroid nucleus even look like at all? So the steroid nucleus is made up of 3 cyclohexane rings, okay? 3 cyclohexanes. What a name I will call 3 hexagons arranged in this manner, and then 1 pentagon attaching to the 3rd cyclohexane ring. Is that okay? Fine. And there's an involving side chains over here, methyl groups. Awesome. So now, what is completing the structure of steroid nucleus is an important component of cholesterol, for which is one of the major reasons why we come to this detailed structure of cholesterol. It is a hydroxide ion. Why is it very, very important? Because it is the only polar section of cholesterol. It is the only water-loving section of cholesterol, and it is determining how the cholesterol molecules in the cell membrane are arranged. So please take note of this hydroxide ion polar section of cholesterol. Is that okay? Fine. So what we have on the board right now is our steroid nucleus of cholesterol. Very, very important. Why? Because all the derivatives of cholesterol are all having this special nucleus of cholesterol called steroid nucleus. So what are these derivatives? Some of these derivatives are the steroid hormones that we have, okay? The estrogen, the progesterone, the testosterone, even the adrenocorticoids like the aldosterone, the cortisol, vitamin D, okay, and even bile salts. They are all derived from cholesterol. For that matter, they bear this steroid nucleus in their structures. Is that okay? Fine. So what is the final thing completing the structure of cholesterol? This thing is binding to the apex of our pentagon or our cyclohexane structure. It's actually an aliphatic chain, okay, over here as you can see over here. So guys, this is our structure of cholesterol. This is the typical structure of cholesterol. So you can imagine if you are to represent about 10 out of this in drawing the foundation of the cell membrane, you can imagine how much time that is going to cost us, right? So it is important for us to deduce much more simpler structure out of what we have on the board right now. So you can use that one to represent our cholesterol molecules wherever that we draw. Is that okay? Fine. So let's try to make this one simpler. So making it simpler and smaller, you're just going to make an outline around what we have on the board right now. Okay, let's make an outline. So this giant oval is representing an outline around our cumbersome cholesterol molecule. So this is going to help us to make it a little bit smaller. So wherever that we see cholesterol molecule, you're going to just consider a long-standing oval which is shaded in the middle. So guys, this is going to represent our cholesterol molecule wherever that we draw it. Is that okay? It's just a long-standing oval which is shaded. Fine. If you remember, we used the molecule to represent a phospholipid structure. Remember, we said it's just what a polar head with two, let's use just a polar head with what? 2 hydrophobic tails. This is representing our phospholipid structures in the cell membrane. Okay. Awesome. And now we know that a long-standing oval which is shaded in the middle is what is representing our cholesterol. Last important fact to bear in mind about cholesterol is that it is also having a hand in the fluidity of cell membrane. And how does it affect it? Increasing the number of cholesterol molecules in the cell membrane decreases the fluidity. It's just like that, so simple. So the more cholesterol molecules you have in the cell membrane, the less fluid the cell membrane. So simple, just like that. So take note of that. Now we have our representative structures, okay, for the cholesterol molecules and the phospholipid molecules. Now, remember we said that there are 3 lipid building blocks for the foundation of cell membrane. So which one is left? If you remember, it's sphingolipids. So it's now time for us to consider our sphingolipid and also know how we can make it small and then use it to represent it wherever that we draw. So let's get into the structure of sphingolipid. Now, about sphingolipids type of lipids in the cell membrane structure, in fact, you don't really worry yourself so much about its typical look. Why? Because they look similar to phospholipids I've already covered. Almost similar. The only thing that is changing is the backbone that you have differently. Okay, in the phospholipid, you realize that there is this backbone called what? Glycerol. But in sphingolipids, there is a different backbone. That is what is causing the change between these two types of lipids in the cell membrane. So what is the name of this backbone found in sphingolipids? We call this backbone sphingosine. Sphingosine. In fact, that is the reason why we call them sphingolipids, because they bear a backbone in the name sphingosine. So guys, because of that, in representing them in a more simple and much more smaller structure, we're just going to consider what we use to represent our phospholipids, okay? The polar head group with two hydrophobic tails, but change the structure a little bit from the phospholipid structures. We're just going to leave a dot in the center of the polar heads of the sphingolipid structures. Whenever we are trying to draw sphingolipids, we're just going to consider this as we have on the board. Okay, so this is going to represent our what? Our sphingolipid structures. Excellent. But there is one thing to talk about sphingolipids, okay? The sphingolipid types in cell membranes are a little bit a lot. But the commonest type found in there is what we call sphingomyelin. It's the commonest type of sphingolipids in the cell membrane structure. That is the commonest type in the cell membrane structure. Even though there are other types of sphingolipids found in some cells, cell membrane, like gangliosides, like cerebrocytes, like globocytes, and all that. But the most common type of sphingolipids that can be found in cell membranes, especially in neurons, is what we call the sphingomyelin. But about sphingomyelin type of sphingolipids, there's one thing about it. In its structure, there is a phosphate ion. So because of that, some literatures consider sphingomyelin to be also a type of phospholipids. Because remember, when we're dealing with the phospholipid structures, the only reason why we term them as phospholipids is because they bear a phosphate ion in their structure. And in sphingolipids type of sphingolipids too, there is a phosphate ion in its structure. For that matter, that is the reason why we consider them to be also an example of what phospholipid structure is, just as simple as that. Take note of that. So when you start with the types of phospholipids that we have, we're going to what? Include sphingomyelin as a type of phospholipids. Awesome. Fine. Okay, mediSars, so this brings us to the end of the lipid components of our cell membrane structure. The next time we'll meet, we're going to do with how we can build a typical human cell membrane with this lipid building blocks. Okay, please, if you found it useful, kindly like, and don't forget to also hit the notification bell, as well as leaving a comment to tell us your positive ideas about how you think we can better up our mode of presentation, and also those encouraging comments of yours to go that sort. Okay, please, lastly, don't forget to subscribe as well. Okay guys, until we meet ourselves there.